Hey everyone, let's talk a bit about organic molecules or the biological molecules that you watched in the crash course video. So we're going to kind of cruise through this since you've been introduced to it a couple of times already with those videos and the chemical reaction video that I gave you before. So a quick recap, we have 4 different types of organic molecules. Now, when we're talking about organic molecules, we're talking about things that are carbon-based. Now, it can't just have carbon to be organic. For example, CO2, carbon dioxide, is not considered organic because it also needs to have hydrogen. So anything that has carbon and hydrogen in it would be considered organic. So the 4 types of organic molecules that we have are proteins, carbohydrates, lipids, and nucleic acids. So let's start by talking about carbohydrates. So if you remember, carbohydrates are basically sugars. So they are classified by the number of simple sugars that we have building these kind of chains of bigger sugars. So what are the functions for your body? Well, it kind of depends. So let's talk a little bit about the different types of sugars. You can have a monosaccharide, and some examples of that would be glucose, which is important because that's your blood sugar, and fructose, which is a fruit sugar, but you also make fructose. We'll talk about that later on. Disaccharides would be double sugars, so they have 2 sugars linked together. Examples of that would be things like sucrose, which is table sugar, lactose, which is found in milk, and my personal favorite, maltose, which you can use for malted milk balls, putting malt on a sundae, or making beer. And then we have really big sugars, which we call polysaccharides. So these are long, long chains of simple sugars linked together. And the 3 different types that I want you to be familiar with are cellulose, which you learned about in the other video a bit. Cellulose is a plant structural polysaccharide, so it gives structure and support to the cell wall of a plant cell. You cannot digest cellulose. You don't have the ability to break it down. Some animals can. Bacteria can break down cellulose. So why then do you need to eat plant material if you can't digest it? Well, keep this in mind. If I cannot digest it, then I can't get energy from it. That is probably one of the most common answers that I get, and it is not correct. You need this non-digestible fiber for a couple of reasons. It helps to keep stuff moving along in your digestive tract. So think of it kind of like nature's broom, sort of sweeping stuff through the tract. And it's going to help you feel full longer, which is going to help you with calorie control, because if I'm full longer, then I don't need to eat as often to kind of keep myself from getting hungry. And then that means I'm not going to intake as many calories. And it slows glucose absorption. So if you have other carbohydrates in your diet, in that particular meal, and I also have these cellulose molecules, these dietary plant fibers, then I'm not going to get as big a spike of blood sugar. It's going to be more gradual so that my pancreas can deal with that. So I need cellulose in the diet, but not for energy. I need it for these reasons up here. Glycogen is an animal energy storage molecule. So you store glycogen in your liver and muscles, and it allows us to very quickly replenish blood sugar. So if your blood sugar drops, you break down glycogen stores and release that back into the bloodstream as glucose. And then we have plant energy storage, which is in the form of starch. Now, starch is very similarly shaped to cellulose, but you can break it down. So this is plant energy storage. You can digest it and break it down into simpler sugars. So you can use that for energy. So if you notice, most of these different substances here, these carbohydrates, are going to be used for energy, with the exception of cellulose. So carbohydrates we kind of like to draw like this, but they also like to form rings. We can link those rings together to make big long chains called polysaccharides. Remember we talked before about the bonds? Bonds are like pixie sticks. The more bonds I have, the more pixie sticks, the more energy I get. So I'm going to jump through this really quick, and because we already talked about all of those types of molecules. So let's talk about lipids. Lipids are hydrophobic. If I'm phobic of something, it means I don't like it, and hydro means water. So these are molecules that don't like water. You can dissolve carbohydrates in water. That's easy. I can dissolve starch, like cornstarch, in water, or sugar in water. I can even dissolve some proteins in water, but I cannot dissolve lipids in water. We have our true fats. So these are the triglycerides. And these are either saturated or unsaturated. We have the steroids, and we have our phospholipids. So let's look at each of those. So if we look at a triglyceride, I always think that this is my triglyceride, right? So I've got my 3 fingers, which represent the fatty acid tails. So those are these long chains here. This is a fatty acid tail. There's another one and another one. And then those are attached to this big glycerol molecule. Now these long chains can all be identical, or they could be different. Notice this one is a little bit shorter than the second one. Notice that this one has a double bond in it, and that makes it bend. So they can all be the same or they can all be different. There's a lot of variability. Now if I have a saturated fat, if I just look at that long fatty acid tail, it's just this long chain of carbons, all with single bonds between them. So when we say that it's saturated, it is saturated with hydrogen atoms. It has a maximum number of hydrogen atoms that it could hold. That means it's going to be nice and flat, and it's going to lay nice and flat with the next molecule. So you end up with something that's very dense at room temperature. So this is a solid at room temp. So these would be things like butter or lard, animal fat. If, however, I remove some hydrogens, then I have to create a double bond so that each of these carbons still has the 4 bonds that it needs. So if it's unsaturated, it means it does not have the max number of hydrogens, and this creates bends or kinks. So if I take, for example, this piece of paper. Every time there's a double bond in it, I form a kink. Now imagine I have lots of double bonds in all 3 of those fatty acids. So I end up with this very bendy looking molecule. If I take another unsaturated fat, so I have to create a bunch of kinks in those chains, now when I try to stack them, they don't stack nice and tight anymore. So these are going to be looser or liquid at room temperature. So these are going to be most of your plant oils. Now, coconut oil is an exception to that rule. It's kind of an odd one, but your peanut oil, sunflower oil, canola oil, and so on. Now, if I want to just be really, really simple with this, we know that butter and lard are bad for us, even if they taste good, and we know that the oils like olive oil are generally better for us. And the reason is fairly simple. If I'm a liquid at room temperature, and then you put me in the body, which is warmer than that, then I'm going to stay a liquid for sure. But if you put me in the body as a solid, even though the body's warmer, I may or may not liquefy. So these saturated fats are more likely to come out of solution, form plaques in the walls of arteries, and potentially form blockages that can be life-threatening. Now my triglycerides, if I'm storing these fats, are going to tend to be used for long-term energy storage. I might use them for some protection. There's a fat pad around your kidneys, for example. If you like to swim, they give you buoyancy, maybe a little padding if you fall. But these are kind of our big main functions. Now you've heard of trans fats. Trans fats are bad. Basically, you take an unsaturated fat and you hydrogenate it. So if you've ever looked at a package of food and it says in the food label, partially hydrogenated vegetable oil, you might think, oh, I remember Drake said oils are good for you, so I'm going to eat some of these things. However, it's partially hydrogenated. So they took these carbons that have double bonds and they forced hydrogens on them, which got rid of the double bonds, made it a single bond again. Same thing here. So then what I did is I turned my relatively healthy unsaturated fat into a saturated fat. In addition, the bonds that are formed are even harder for your body to break down, and so it's a little bit like this. If you eat a saturated fat, I always think it's kind of like inviting a serial killer over for dinner. Now, you might have a nice dinner, interesting conversation for sure, but there's a good chance that maybe he'll leave before he kills anybody. However, if I eat a trans fat, it's harder for my body to break down. So it's actually like I invited the serial killer to move into my basement. So now he's living there for a few months. There's a better chance that at some point maybe he's going to snap and kill us. So you want to avoid these guys. Steroids have this general shape. They kind of have these little blocks that are all linked together. What I really want you to know about steroids is this. I want you to know that they're lipids, which means they're hydrophobic. They don't like water. That's going to be really, really important when we talk about how steroid hormones signal cells. So we'll talk about that more when we get to the endocrine system. You should know some examples of steroids. So those would be things like your sex hormones and cholesterol. Cholesterol gets a bad rap, but it's actually important as a carrier, as a component of cell membranes, and as a building block for making these hormones. And we'll talk more about steroid hormones later on. Phospholipids look a little bit like a triglyceride. So here's a triglyceride, right? So there's my fatty acids, my three fingers sticking out. What if I take one of those away? So now I take away a fatty acid. This third carbon down here is going to be unhappy. So I'm going to attach a phosphate portion to that fatty acid. Now, the fatty acid tails, they still don't like water. So these are hydrophobic still. But this part, the phosphate group, likes water. So it's hydrophilic. So we're going to take these phospholipids and we're going to put them like this together to form a phospholipid bilayer membrane, like this. So we zoom in on this cell membrane here. You've got the inside of the cell, you've got the outside of the cell, and then you've got all these little hydrophobic heads or hydrophilic heads facing the water, and then you've got all of the hydrophobic tails in the middle. And that's going to be important for helping to regulate what goes in and out of the cell. We'll talk about that more when we get to cells. So let's stop at this point, and we'll start up another video and talk about proteins and nucleic acids.